Hi everyone, my next topic is motion perception. Here are the learning outcomes. We'll take a look at what kind of information motion provides to us. We'll narrow down a little bit the brain regions that handle motion perception. I will go into one proposed type of motion detector called a Reichardt motion detector. We'll see that it's missing one important piece of information, which is totally given away by number 4. We need to take eye movements into consideration, and this brings up something called corollary discharge theory. We'll explore some apparent motion or stroboscopic motion, and we'll take a look at some factors that influence perception of this type of motion illusion. I will introduce a theoretical problem called the motion correspondence problem and one proposed way of overcoming it. I did mention biological motion in a previous lecture. I will now explain biological motion and give you some examples, and also one implication of this called the uncanny valley. And then number 8, because I demanded it, how do we perceive motion pictures? But first, we need to take a look at the information provided by motion. In other words, really, why do we have motion perception in the first place? So in no particular order, motion grabs our attention. If there's an otherwise motionless scene, if there's a bit of motion in there, it's definitely going to attract your attention. And that is adaptive. That's important. It has survival value. Motion can also tell us about the three-dimensional shape of an object, and this will help us identify what that object is. I have an example for you coming up. I can't embed it in this exact slide. I'm having some problems with the software. But it will actually show you a motion illusion called the hollow face mask illusion. As you watch it rotate, it'll seem to flip back and forth from convex to concave and then back again. That's a bit of a trick that really shows an interaction between motion and shape. Along the lines of the hollow face mask illusion, I want to introduce Thinky the Dragon to you. The cool thing about Thinky is you can make one yourself. I have some links on my website. You can print it out. You'll need to print it out on some pretty stiff paper, but you print it out, cut it out, and stick it together with about 1,000 pieces of tape, and you too can make a Thinky the Dragon. What does Thinky do? Check out this video.
Here's our little dragon, sitting on the window ledge. But look what happens the minute we move. His head twists and turns to follow us as we move around the room. It's all an illusion, of course. He's not really moving at all. It's a version of the hollow face illusion, but it's a very powerful effect. And not just side to side. If we move up and down, again his head cranes to follow us. But our brain has made an assumption about the structure of the dragon's head. And if we move far enough around to the side, we suddenly realize the structure is different and the illusion fails.
Some more things that motion does for us. Motion helps us separate figure and ground. Let's see if this can work. Can you see the object in there? And no, the object isn't a scribble. Hey, I think I see it. No. Let me try to add some motion in there. Oh, there's a bird in there. Let me reset it and show it again. And again. And again. Now, if it's not moving, it's kind of tough to see the bird. Maybe you think you can see the wing, but it's tough to see the entire bird. But when it's moving, then it's really easy to see that bird. And last but not least, motion allows us to interact with the environment. We're not plants. We move around, we locomote. It's important for us to be able to see things even when we are in motion too. I want to take a look a little bit at the brain regions that handle motion perception. Now, I did already talk a bit about a subset of the brain that handles motion. Do you remember the magnocellular layers of the LGN? Does that system really handle motion? And that was answered in this study by V.S. Ramachandran and Richard Gregory. But they didn't approach it with scalpels and microelectrodes. It's a pretty clever solution to trying to answer this question. Instead of cutting anybody or any animal open, they used a really unique class of stimuli called isoluminant. Iso means the same. So isoluminant stimuli are stimuli that have the same luminance, that is, the same amount of light. So there are no differences in lightness within these stimuli, but they do differ in terms of hue, that is, wavelength. Now, if you show someone these stimuli, you ask them what they see, well, they can see it, and they'll tell you, I see lines or stripes or whatever. But then you start the stimuli moving. And the thing about it is, one part of your brain says, well, it's moving because at time 1, it's over here, and then at time 2, it's over there. So over here, and then it's over there. Okay, so it must be moving. But your eyes don't really agree with that. Your eyes tell you this thing is kind of frozen, and then it seems to stutter and then skip ahead. It does not produce a smooth percept of motion. Now, it's really difficult to show you isoluminant stimuli because I would have to calibrate my display, first of all, and I would have to calibrate it for each of you, which is, I can't actually do that, so I'm going to try the next best thing. If this doesn't work, well, it's not me, it's you. So this is called the stepping feet illusion, and it does kind of depend on your monitor and exactly where you're sitting, so don't sit too close. So pretty clearly what we've got is a dark blue block and then a light yellow block. And these two rectangles are moving smoothly across your screen from left to right. But when the black and white stripes appear, those blocks seem to take little steps, kind of like they're going ee-oo, 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 ee-oo. And they seem to go back and forth, like sometimes they're stepping and sometimes they're moving smoothly. Well, in reality, the whole time they're moving smoothly. But when the vertical black and white lines appear, the dark blue rectangle seems to blend into those black lines, and the yellow square kind of blends into the light gray lines a little bit. This is the best I can do to show you isoluminant stimuli. We can conclude from this that the magno system handles motion. Now remember, the magno system is not sensitive to color. It's the parvo system. When there are differences in luminance, the magno system has no problem. But remember, these stimuli are isoluminant, so the magno has a hard time kind of grabbing on to where exactly those stimuli are, and we get a really impaired perception of motion. What we're doing is we're relying on our parvo system, which does not do a great job in processing motion. This is a very clever experiment using carefully designed stimuli to make some conclusions about parts of the brain, again, without cutting anybody open. Nice. Now we can go from the brain and try to go to a lower level. Now the brain is not detecting motion, probably. It's probably being detected in the eye. But motion isn't the same thing as color. How does motion actually get detected? Now there have been a number of proposals over the year. This is one of the best, proposed by Werner Reichart, and it's called the Reichart Motion Detector Circuit. I have to explain what you're looking at here a little bit. So we have these funny kind of shapes, A through F. Those are intended to be receptors. So they could be rods or cones, and they're pointed at different locations in space. They connect to cells G, I, and K, and those cells connect to their neighbors. So it looks like they have a T-shaped ending, which means inhibition, lateral inhibition, because they're inhibiting their neighbor off to the side. Cells H, J, and L will pass on their activity only when motion is occurring to the left. Only when motion is to the left. Let's take a step back and see what happens when motion is to the right. That would mean a point of light starting here at A, then hitting B, then C, and then finally reaching F. That motion is to the right. Now what first happens is receptor A gets activated, and its activation will travel along slowly down to G. But at the same time, the light has now moved on, and it's stimulating B, which sends its activity down to H. But what happens is we get this inhibitory signal from G just as H is getting the excitation from B, and that will cancel out. The same thing happens here at J. The activity from I and D arrive at the same time, cancel each other out. Same thing here at L. From K and F, they cancel each other out. As a result, M does not change its firing rate. However, if the motion is to the left, starting at F and moving to A, the timing is all different. F gets activated, sends its activity to L, and L starts to get activated. Now by this time, the light has struck E and the signal is traveling down, E reaching K, but K releases its inhibitory neurotransmitter too late. L has fired, and it's already sending its activity down to M. The same thing happens at J and at H, and all of this activity converges at M, and it causes M to fire. What does M do? M signals motion to the left. It will not be activated by motion to the right. So the key through all of this is the timing. The timing has to be just right in order for motion to be detected in one direction and not in the other direction. Now I know that's probably a bit hard to follow with me pointing to things and trying to use my mouse here. I have an animation of this which is even simpler than this. It's really oversimplified, but I have it, so I'll show it to you. Here it is. Okay, you'll notice in this circuit, this neuron fires when motion is to the right. When motion is to the left, it does not fire. So this is the opposite of what I showed you on the previous slide. And it's way, way, way oversimplified. No, it doesn't depend on the length of the axons, but this is the best animation I could find. Now the Reichert motion detector seems to be pretty clever. It's describing, using known structures, how motion might be detected within the retina. That's super cool, except there's a couple of problems. Here are the two cases that are problematic. The first one is if you are pursuing an object. It is called pursuit. Let's say you're at a racetrack and you're watching a car go around the racetrack and you're focusing on that one car. It's falling on your fovea, and you are moving your eye to follow that car around the racetrack. Now the car is definitely moving at over 100 kilometers an hour, but it's not moving across your retina. In this case, it would not activate the Reichert motion detector because the position of that car doesn't change across your eye. But yet we still know the car is moving. How does that work? Or, kind of the exact opposite, if you look at a stationary scene, look at the room around you, hold your head steady, and just move your eyes back and forth. Well, now the entire scene is swooshing, sweeping across your retina, so that should be activating a whole bunch of Reichert motion detectors, so you should be experiencing, oh, the room is swooshing back and forth. No, of course it's not. You know that the room is motionless. So what is the common factor in these two cases? It's the motion of your eyes. We need to actually take that into account to fully explain and account for motion perception. So the Reichert motion detector was a good first crack at it, but it wasn't complete. So to pick up where it left off, we have something called corollary discharge theory or corollary discharge theory. Take your pick. This theory has 4 important components. One component is the image displacement signal, and it's some kind of mechanism that detects motion across the retina. What kind of proposed neural circuit might we have to detect motion across the retina? Yeah, this could potentially be the Reichardt motion detector. So the IDS could be getting its signals from something very like the Reichardt motion detector. Okay, we have that. Next is something called the motor signal. Now this is information sent to the muscles of your eye. This is how your eyes move. Okay, so far so good. The next component is the corollary discharge signal, and this is what gives this theory its name. The CDS is a copy of the motor signal. So the motor signal goes from a part of your brain to your eyes. The corollary discharge signal goes presumably from that same part of your brain to another part of your brain. Where does it go to? Okay, that's the fourth component, and that's called the comparator. The comparator kind of calculates motion perception. I know that sounds a bit strange because you might think we just passively pick up the motion of stimuli, but it may not be that way. It might be that it has to be computed, calculated, or processed. Now, according to this theory, the comparator will signal motion under one of two conditions. When we have an IDS, image displacement signal, or a corollary discharge signal, but not both. Importantly, not both together simultaneously. So let's just take them one by one. If we have an IDS but no CDS, what that means is there's something moving across your retina, IDS, but your eyes are not moving, no CDS. Second case is there is no IDS, but there is a CDS. That's kind of like the example of pursuit. I'm looking at this car going around the racetrack, but it's not moving across my retina, but my eyes are moving to follow it. So what about the third case, when we have an IDS and a CDS? What would that be like? Well, that would be like the case of you holding stationary, moving your eyes back and forth across a stationary scene. You get an image displacement signal, the image swooshing back and forth across your retina, and there's also a corollary discharge signal because your eyes are moving. What happens in this third case is the IDS and the CDS cancel each other out, and you correctly do not perceive motion. You move your eyes back and forth across a stationary scene, you know that the room around you is not moving at all. So in talking about the Reichardt motion detector, I've already covered the case of the IDS. I want to turn to the case of having motion perception with the corollary discharge signal. So I want to go over 4 possibilities. Some of these you can try yourself, and some of them I would not recommend that you try. Number 1, you can try this yourself. So what you have to do is very briefly look at something pretty bright. This will produce an after image. Okay, so you'll have to be in a completely dark room, open your eyes, and really briefly shine a flashlight into your eyes. Don't get a super bright one. I don't want you damaging your vision. Just do this very briefly, and then turn off the light. You'll be in a completely dark room, and you'll have kind of a purplish spot in your eyes. And if you move your eyes around, that purplish spot will seem to be moving. Well, it's not. It's on the same part of your retina, but your eyes are moving, producing a CDS, and this will seem to be moving around the room. The second one is one of the ones I don't recommend. What you can try to do is, push on your eyeball. But don't push your eyeball out of its socket, okay? What you kind of have to do with your eyeball is push back on your finger. Now, again, you don't want to jostle your eye around, but you want to put some slight pressure on your eyeball such that your eye has to push back against your finger. If you do this correctly, your muscles pushing back will initiate a CDS, and even though nothing is actually moving across your retina, it'll seem like the scene in front of you is moving. Number 3 is one I've talked about already. It's pursuit. There's a moving object and your eyes move with it. So tracking a car, for example. The last one, again, is something that you shouldn't probably try. If your eyes are paralyzed and then you try to move your eyes. So again, don't do this at home, people, okay? Now, if your eyes are paralyzed, try as hard as you might, you cannot actually move your eyes. Now, this means the retinal image is stationary. There's no IDS, but your brain is continuing to try to send messages to your actual eyes, but it's also sending that CDS. It's a bizarre phenomenon in which the person is like lying there. Their eyes are paralyzed. Their eyes are not moving. They're like staring straight ahead. And you say to them, okay, now move your eyes to the left. And they'll try to do that. And their eyes won't move, but they'll go, whoa. And you'll go, what was that all about? Well, the room was like swooshing back and forth. Okay, move your eyes the other way. Whoa. And again, they will experience the room as moving, but ironically, nothing is moving. The room is not moving, their head is not moving, their eyes are not moving, but they're getting the perception of motion. Crazy! That leads me into motion illusions, like stroboscopic and apparent motion. Now you might think this is some kind of special case and it almost never happens. You would be wrong. If you ever experience motion when you're looking at a screen, any kind of screen, it's an illusion. Computer screen, TV screen, movie screen, your phone screen. If you see anything moving on that screen, it's an illusion. The only thing that screens can do is show you static images one after another. But your mind can stitch those together and produce the experience of motion. The only things that can really move are real objects in the real world, physical, tangible things. Let's take a look at the beginnings of the study of apparent motion. And we go way back, over 100 years ago, to a very simple display like this. This is an example of apparent motion. So there are 3 circles on the left, and they seem to be moving to the right. Very simple. And this is the kind of display that Max Wertheimer was interested in. What he concluded in 1912 was that the experience of motion depends on timing. And the specific timing variable is called the interstimulus interval. So in this display that I have here, this rectangle, it's super simple. So I have 2 different displays. One display has the 3 circles on the left, and then they go off very briefly, and then they're replaced by the second display, which is the 3 circles on the right. So I actually have a very short amount of time between those 2 frames of this very simple movie. Wertheimer did a bunch of experiments, and he determined that if the ISI is less than 30 seconds, the stimuli seem to be on simultaneously. At about 60 seconds, you get a continuous experience of movement. Anything greater than about 200 milliseconds now, the stimuli don't seem to belong together. You would see 3 circles on the left, and then 200 milliseconds later, you see a bunch of circles on the right. But it's not like the circles have moved from the left to the right. They seem to be just 2 independent sets of stimuli. Now, Wertheimer wasn't wrong about this. The problem is he wasn't right enough. There are some other factors that are also crucial to the experience of motion perception, and they include the distance between the stimuli. So those circles didn't have far to go to move from one side of the rectangle to the other. But if I had them try to move from one side of the screen to the other in that same amount of time, it wouldn't have produced the same experience of motion. We're kind of asking those circles to move too far in that brief 60 millisecond window of time. The frame duration is important, so how long am I showing you those dots for? If the circles are on the screen for too long, without moving or changing, you kind of get the impression that, oh, these are unmoving circles. So they can't be displayed for too long. And there is another important timing variable, which Wertheimer was close, but it's a little bit more complex than the ISI. The more important variable is called stimulus onset asynchrony, or SOA. This is a bit of a diagram that explains and illustrates SOA. SOA is the sum of the frame duration and the ISI. Now, what these two kind of bumps here represent are the first frame, which is on for a while, it goes off for a while, there's the ISI, and then the second frame comes on for a while and then goes off. SOA turns out to be more important than ISI, so much so that there's something called the SOA law. Now, it is possible to change the frame duration and the ISI without changing the SOA. The SOA is constant, and you can do that by having, say, a shorter frame duration and a longer ISI, or vice versa. You can change frame duration and ISI, but if you don't change their sum, the SOA, you will not change motion perception. That's how important the SOA is. Now, the thorny theoretical problem when it comes to motion perception with these very simple displays of dots, which are admittedly very, very simple. We should be able to wrap our arms around this understanding of motion perception with dots before we move on to more complex things. But even simple displays of dots introduce problems, and that's the motion correspondence problem. So I showed you 2 frames, 3 circles on the left, 3 circles on the right. But how did you match those circles up? There's a whole bunch of possible alternatives, and no, I haven't drawn them all. One possible is that this top circle on the left, when it reappears 60 milliseconds later, has moved over and down, and these 2 circles have moved over and up. Okay, that's a possibility, I guess. Another possibility is the central dot has moved straight across, but the top and the bottom ones are crossing over. Anybody experienced that? No, I didn't think so. And then there's this possibility, which is the one that probably every one of you experienced. But why this one and not these other 2, which are logically possible? Is there some underlying logic to this? So if we have 3 elements, there are 3 factorial possible interpretations. So if we were to have, say, 10 dots, oh my gosh, there are a lot of possible interpretations. But how does our visual system choose the right one every time? And it basically does choose the right one every time. Okay, the good news is there is actually a solution to this, thank goodness. It's called the nearest neighbor principle, as described by Professor Mike Dawson, who is a professor in the Department of Psychology here at the U of A. The one alternative that is chosen is the one that mathematically minimizes the overall traveling distance of the elements. What now? Let me go back to the previous slide. Okay, so we have these, I've drawn these as arrows, and I've done that for a reason. Maybe they're vectors, okay? So a vector tells you what direction something is moving in, and the length of the vector represents how quickly it's moving. So for this dot to reappear all the way down here at the same time with these 2 other dots, it would have to be actually going faster because it has to cover more distance. So we can take a look at every one of these different alternatives, all 6 of them, and pick the one that has the overall shortest vector length. Now this one has 1 big long vector and 2 medium ones there, 1 really short one and 2 really long ones, and look at this one, 3 very short vectors. Which alternative is selected by the visual system? The one that minimizes the overall traveling distance of the elements, and that's why it's called the nearest neighbor principle. Each circle in frame 1 moves to its nearest neighbor in frame 2. What an elegant solution using mathematics to come up with an answer, except there are more problems. You can come up with displays that are more complex. So in this one, in frame 1, okay, it kind of looks like an octagon, but let's pretend that it's a filled circle, okay, in frame 1. And in frame 2, wait a minute, there are 2 elements. On top, on the right, there's a filled circle, and then below it is an unfilled circle. How do we match the elements in frame 1 to the ones at frame 2? So again, there's a number of possibilities. It could be that the filled circle just moves over and up to the right, and then the unfilled circle just appears out of nowhere. Or maybe it's like a nuclear fission or something, that this one particle splits apart into 2 particles. How do we make matches in cases like this that are more complex? Okay, there are answers. When we have cases like this, matches are made based on intensity, not necessarily shape. Okay, now the shapes happen to be common throughout all of this, but if I had, say, a filled circle on the right at the top, and then a filled square at the bottom, well, that's a different shape. No, intensity comes first. The filled circle seems to move over and up to the right. And the unfilled circle, who knows where that's coming from. If there aren't any intensity differences, our visual system next relies on texture. So imagine some of these circles have vertical stripes and some of them have horizontal stripes. Their overall intensity is the same, but now we'd make a match based on texture. And no, I'm not going to go over the entire list of what exactly do we use to make matches. No, that's not the point of it. The point of it is, yeah, there are regularities in the visual system, but memorizing the exact order and rules of it is not what we're going to do in this course.